All right, we're talking about cancer today and how you inherit cancer, and if you can inherit cancer. In general, cancer is just any cell that has lost control of its cell cycle. Any cell that is inappropriately dividing is technically a cancer. Now, that's not usually what we mean when we say somebody has cancer, because there's actually cells that are losing control of their cell cycle all the time. Now, if they do, oh, look at that. Here, we'll just throw everything up there. If a cell does lose control of its cell cycle, most of the time there's checkpoints that tell the cell, I'm inappropriately dividing, and that cell will do one of two things. It will either apoptose, in that it'll start dividing inappropriately, and the cell or neighboring cells will actually recognize it and induce that cell to just commit suicide, right? So cells will often, if they lose control of their cell cycle, just take care of themselves, and you'll never even know that you had a cancer, right? You had inappropriately dividing cells, but they just kill themselves and they completely go away, right? Now, other cells might detect them. So apoptosis would be if the cell itself recognizes that it's inappropriately dividing. Other cells may recognize that a cell in a tissue is inappropriately dividing, and that will elicit an immune response. And you can actually have macrophage cells come invade that tissue, identify the cancerous cells, and engulf them and digest them away. If the immune system detects that, then you're no longer going to have a cancer, and you'll never know that you had the cancer. Those cells will actually just go away, right? So these cells are doing this all the time. It's only when cells have made it past the immune system or are no longer kind of self-regulating themselves that we actually say you would have a cancer, right? That's when it'll be detectable. You'll start growing inappropriate cells. You'll start getting a tumor, and then it might start impinging on function, right? And at that point, we would usually diagnose people with cancer. Yeah, Will?
Just repeat what you just said. When do we say it's cancer?
Well, we would say it's cancer. Most of the time, you actually have inappropriately dividing cells, but we never know it, right? Because the cells just go away. Mm-hmm. If you're diagnosed with cancer, that means it's gone past this point, right? So the cells haven't recognized that they're cancerous and are continuing to divide. And the tissue, the body's immune system, for whatever reason, is not recognizing those as inappropriate. And so that was when you would actually be diagnosed with a cancer, right? Okay. It's a tricky thing for the immune system to recognize cancerous cells, because the immune system is kind of rigged to detect foreign invading cells, right? And so to recognize your own cells is a dangerous business, right? You want to recognize cancerous cells and kill those off, but you don't want to kill off any other cells in your body, because now you have an autoimmune disease, right? So this tension of non-self recognition and when a cell is actually appropriate for the body to kill off, it's a tricky thing. Causes of cancer. There's multiple causes. I'm going to talk about mostly the genetic causes or the genetic underpinnings behind getting a cancer. So I'm going to talk about today 3 different types of genes. These are normal functioning genes in your body, right? But when they have mutated or when they're not functioning appropriately, then they might give you susceptibility to having cancer. And there's 3 main classes of them. Number 1 are called oncogenes. Number 2 are called tumor suppressor genes. Oncogenes are typically genes that trigger a cell to inappropriately divide. Tumor suppressors usually are the kind of checkpoints. If it's inappropriately dividing, does the cell recognize that that's an inappropriate step to take? So one's kind of a positive mechanism, the other one's a negative mechanism. And then the third are called mutator genes. These are genes that are involved in DNA repair. You can imagine if your DNA repair genes are not functioning properly, then you're more likely to accumulate mutations, and you're more likely to get mutations in oncogenes or tumor suppressor genes. So there's a third level of genetic underlying. Having mutations in any of these does not necessarily mean you have cancer, and we'll talk about the interplay of this, but these are underlying genetic mechanisms that are involved in it, right? They may or may not actually mean you have cancer. The second class of causes are some environmental cause. There's actually some schools of thought that say all cancers are either bacterially or virally induced, and that the actual genetic underpinnings are just giving you a proclivity to having cancer, but you wouldn't actually have cancer unless there's some type of stress caused by an infection. Now, there's other camps that say no, it really is sometimes just caused by genetic mutations. So, actually determining exactly where and when and why a cell decided to inappropriately divide, it's still a mystery in a lot of cases, right? So, viral infection is often a case, right? A virus's job is to infect a cell and get the cell usually to reproduce the virus itself. But often viruses will invade a cell, stimulate the cell to start making lots of copies of the virus. But then a helpful thing would be for the virus is to trigger that cell to divide. And so now I've got two cells that are virally infected making more virus. So oftentimes viruses will induce cells to divide and will like back off cell cycle regulation and try and get cells to inappropriately divide. Hepatitis B causes liver cancer. There's ovarian cancer, the papilloma virus, excuse me. That one was in the news just a couple of years ago. There was like some states that were trying to force vaccination on all females for the papilloma virus. So we know some really specific viruses that really do. The virus infects and it almost always causes cancer. But sometimes the virus might stimulate the cell to try and grow, but the cell itself knows I shouldn't grow, and so even though the virus might be trying to get it to inappropriately divide, the cell might have the capability to not bend under the influence of the virus, right? Okay, so I'm going to step through these three genes, but let's talk about tumors first. So this is a lung cancer cell. This is normal lung epithelial tissue, and this is in cell culture. So people have gotten normal lung epithelial tissue and then taken some of the tumor cells out. And so this is a lung cancer here. This is actually a derivative of the lung tissue, right? This cell is actually looking very different from the other lung tissue. So it started off looking as one of these very flat epithelial-like. And in this case, the cancer has totally changed the shape and function of this cell. There's two classes of tumors. So you may have inappropriately dividing cells, right? They've lost control of their cell cycle. They're dividing inappropriately. They're causing a tumor. But that tumor we might call benign, right? It's cancerous. But benign tumors don't usually pose too much of a health problem unless they start impinging on an organ's function, like blocking a cavity that's supposed to be open, or blocking a valve that's supposed to leave access. Most of the time, benign tumors don't cause much damage. It's because they have the same characteristics as the tissue of origin, the organ of origin, right? If you've just got a couple of extra, prostate cells, or a couple of extra, cells in the colon, right? This is a benign colon tumor here. So this is a colonoscopy. This is the inside of the colon, and then we find this little guy called a polyp. It's a little mass of cells, but they mostly have the same characteristics as the tissue of origin, right? They're still kind of doing like secretory things and trying to absorb material. It's kind of still acting like a colon cell or like a colon tissue, and they grow really slowly typically. These are usually not harmful. If you find them, and in regular colonoscopies, if they find them, they cut them out and remove them. If it's impinging on some organ's function or it's causing discomfort, you can go in and remove a benign tumor. But if it's not, you may just leave it. It might grow so slowly that it's never ever going to pose an actual threat. Now, the really difficult, the really harmful are malignant tumors. A malignant tumor is a cell that's inappropriately dividing, but it's now taken on different characteristics than the tissue that it originated in. So it's going to play some other role, and it's not going to just happily sit there and slowly grow. Oftentimes, malignant tumors have very rapid growth. And it's really dangerous if that cell starts migrating to other tissues in the body. So a malignant tumor that is moving, we call it a metastasized tumor. Now, most cells in your body aren't migratory, right? Your skin cells just sit in your skin, right? They don't move any place. And your liver cells just sit in the liver and they don't move any place. Same with your lung cells, right? So most cell types, to become motile is a fairly hard thing to do, right? You have to turn on genes that are necessary for making filopodia, and you have to break contact with the other cells that you used to be sticking to and be an individually self-sufficient cell. Most tissues in your body can't do that, actually. Most tissues, if they stop making the cell-cell contacts with the other tissues, they will just undergo apoptosis. Most cells are actually kind of, in a sense, on the verge of killing themselves. And the only thing that's keeping them alive is positive reinforcement from the tissue around them, right? If they're appropriately sticking to their neighbor, then that is sending signals to inhibit apoptosis. And so oftentimes, actually, when you try and grow cells in cell culture, this is especially true of brain cells, but other cells in your body as well. If you try and grow brain cells in a petri dish, they almost always die, right? You have to put in signaling molecules in the media that tell those brain cells, trick them into thinking that you're actually still in a brain, right? Don't die. But if you just try and grow them in just normal growth media, just nutrients, they will just die, right? So you have to convince the cells that they're really in the same place that you took them out of. So most cells, that's kind of a checkpoint for malignancy and for metastasis, right? You could be inappropriately dividing as long as you're sticking to the surrounding tissue. But as soon as you broke off from the other surrounding tissues and tried to metastasize, you might just die, right? So there's several levels that a tumor would have to go through to become malignant. Now, sometimes benign tumors, like ones in the colon, can become malignant and can metastasize. So it's often a good idea if you find these to remove them, right? They're not harmful in themselves, but maybe later on they're going to become harmful. This is actually a metastasized colon cancer. And instead of just forming, just growing inappropriately in the location that they were, so these polyps are usually on a little stalk, right? And so the cells started inappropriately dividing and they just kind of made this column mass. But here, these cells started in one location, but they're actually now spreading along the sides of the cells, right? They're growing and moving now. They've actually dislodged themselves and then can start migrating around the body. Cells that are normally motile are really dangerous if they become cancerous. This is why leukemia and bone cancers are really bad, because bone marrow cells, that's their job, or they turn into red blood cells. They turn into the kind of cells that normally migrate all through your body, right? And so cancers in your lymph system or cancers in your bone, those cells are already kind of pre-primed to be motile. So if they become cancerous, you're in trouble because they've got a much higher incidence of becoming metastasized and being capable of moving around the body. Right? So tissue of origin is really important when we think about how bad is this cancer is the cell type going to be if it got cancerous. Right? All right, questions about benign and malignant tumors? Yeah.
So if the cancer moves, is it able to attach then anywhere it's moving to? And when you say move, I feel like I'm thinking like traveling to another city in some sense, like they're just cruising around in a liver cell cancer, or something, go to the lung and start. Yeah.
So like this little guy. So this is a normal lung cell, this epithelial, big flat cell. It's coating the inside of the lungs. What this cancer cell has done has rolled itself up into a ball, and then notice all these tiny little appendages coming off of it. This guy is primed and ready to start moving. He is a metastasized cancer because he can reach out these appendages and kind of roll himself along and start moving around and move outside of the lung tissue. So I'm going to like give this a purpose in like teleology, but like if a cell becomes motile, it's moving around, and most of the time what's doing is kind of looking for a place that's got more nutrients. One of the things that usually keeps a cell from being able to become really rapidly growing is just a lack of nutrients in the area, right? If you start growing a polyp here, cells way out at the tip here are pretty far away from any vascular system, right? And so how are they going to get many nutrients to actually get the mass and get the energy to divide more? They usually can't. They're pretty slow growing. But if a cell has actually become motile, it actually moves around the body and will tend to stick to places where there's lots of vascularization, and they can get more nutrients and they can grow bigger. So I'm giving that like cancer cells have purpose and they're trying to go do that. But that's a big factor in how rapidly you grow are the nutrients available. So.
Is it only part of the cancer that moves, or is it that whole?
Yeah, well, sometimes cancers tend to stick together, and some other cancers tend to dissociate into single cells and just go all over the body. So the tissue of origin makes a big difference. Yeah, if it's a metastasized colon cancer, I mean, the first thing it's going to do is just migrate around and stick to tissue that it knows. But this could eventually, a single cell might separate off and get into the bloodstream and migrate around and stick to other tissues. If you're lymph and if you're bone marrow, though, you've already got prime access to going everywhere in the body. And so those will tend to be more dispersed, and single individual cells will go further. So it depends on the tissue of origin and what kind of mutations and what happened in its life cycle. When did it? I mean, the longer you let it go, too, the more likely it's going to differentiate even further from the tissue of origin and be able to move outside of it. So, yeah. Other questions? All right, so I'm going to step through the different gene types. Oncogenes so oncology is the study of cancer. Oncogenes are genes that cause cancer. Most oncogenes start off as being very normal, useful genes in the cell, right? All of your genes have a normal function to them, right? And so oncogenes start off as being what we call proto-oncogenes. A normal functioning allele, wild-type allele of a gene, when mutated, if it becomes an oncogene, this is the allele that would actually cause the cancer or contribute to the cancer, we would call that allele an oncogene. A normal one that's just playing a normal role in the cell, we would call a proto-oncogene, right? It has the potential to become an oncogene, but it's just doing its normal function, right? These are often overproduced genes, and they have a dominant effect. So normally they'd be doing some normal cell work. If you start overexpressing them or overactivating them, they could then trigger the cell to uncontrollably grow. So genes that are proto-oncogenes are typically genes that stimulate cells to divide. And here's a little signaling cascade pathway. Most cells, if they're going to divide, they don't just spontaneously divide on their own. They're usually signaled by some other tissue or some other hormone to actually divide. So here's some growth factor molecules that have been secreted by some other tissue. Growth factor molecules would hit a receptor in the membrane of this cell. That receptor would have some signaling cascade pathway, some mechanism by which you can tell that a growth factor actually hit it. Something would end up turning on new genes in the genome, and then you produce proteins that actually stimulate the cell to divide, right? Turn on the making of more microtubules and start, making a spindle and going through G1 and S phase and replicating your genome and doing all the things to turn on cell division. So an oncogene actually could be any overstimulation at any point in this process, right? You could have overproduction of growth factors, right? So if you've got a gene The cancer might actually not be a mutation in the gene that's actually becoming the tumor. You might have a mutation of an enzyme in another cell, right? So another cell's got a mutation, and that mutation is causing it to make too much growth factor, right? So if you've got some other cell that has a mutation, but what that mutation is doing is dumping a bunch of growth factors into the area, that cell then might be telling other cells to inappropriately divide, and they themselves might not actually have the oncogene, right? Now you could have receptors in a cell that are defective too. You might have a mutant receptor that thinks it's being stimulated by growth factors, but when actually there are no growth factors around, right? It's got these, phantom growth factor signals on it. It thinks it's been turned on, and so it turns on genes that stimulate the cell to divide. Yeah. You could also then have mutations in promoter regions or transcription factors, and so these transcription factors have to get turned on to turn on the proteins to make the cell divide. So if you've got inappropriately acting transcription factors because the transcription factors have mutated and they're sticking inappropriately and are turned on too much, then you could have an oncogene going directly here, turning on more proteins to stimulate cell division. So at any point in this pathway, you have the potential to have an oncogene, right? Questions about these guys? I say this acts in a dominant manner because if one of the alleles has become mutated and is an oncogene now, it doesn't matter what the other allele was, right? An overstimulation, you only have to have overstimulation in one gene to get this signal going. Usually, one gene mutation, one oncogene mutation is not sufficient to cause a cancer, right? Because there's checkpoints in it as well, right? The cell has checkpoints to make sure. So you might get one inappropriate growth factor signal, and it might turn on new proteins that stimulate cell division, but there's other checkpoints in the cell that say like, oh, that doesn't seem right. Like, I'm sticking to my neighbors and I'm doing all these good things that say I shouldn't divide. One single growth factor may not be sufficient to get you to turn into a cancer. So you typically have to have, it's usually less than two. You could have two mutations in proto-oncogenes that give you inappropriate stimulation, and you probably that cell probably won't become cancerous. But if you have three or more, three or some other stimulus, then you might actually have cancer, right? So cancer's tricky. It's not like you just have one mutant allele and boom, your cells become cancerous. It's kind of a domino effect or a buildup. Now, some of these control levels are due to the second type of gene. These are called tumor suppressor genes. These are genes that make proteins that normally repress cell division or monitor the cell's cycle to make sure that it's not overgrowing for its conditions, right? So these are going to be genes that inhibit cell division. Now, it could be a very similar pathway that these tumor suppressor genes are working in. There could be receptors in that cell's membrane that actually receive growth inhibiting factors. Cells usually want to be in their proper location and not in another location. There's usually two kinds of signals going on, right? So cells are usually sticking to each other, and that just cell-cell contact is often a repression factor, right? Sticking to this cell type says don't divide anymore, right? And then also, if a cell comes loose, not only does it have that inhibiting apoptotic effect, but if it migrated to a foreign tissue, that foreign tissue might actually be secreting signals that tell inappropriate cells, you're not welcome here, right? So if you actually do certain transplants, you try and transplant certain tissue into a different tissue in the body, that tissue will just die because it's no longer hooked up to the tissue that it used to be hooked up, and it's also getting unfriendly signals from the other tissue, right? So one type of signaling factor might be telling a cell in one tissue, yeah, this is a good place for you to be. So like in a liver cell, and I'm just making this up, I have no idea if this is true, but like think of a liver cell. A liver cell sends signals out to its cells saying, we're the liver. And everyone who's a liver cell recognizes that signal, says, oh yeah, I'm in the right place. Right? You take a skin cell and you put it in the liver, that liver signal might actually stick to a receptor in that skin cell, and that might say, you're in the wrong place, right? A happy liver cell signal is not a happy signal to a skin cell, right? It's going to kill it off. It's going to trigger that to either apoptose, or at least you're not in a place where you should be dividing, right, or contributing to our tissue, right? So you can have growth inhibiting factors hitting receptors. Those receptors turn on transcription factors and turn on proteins that stop the cell from dividing, right? So this is going to act in a recessive manner. So if you have a mutation in one of the receptors, you can live with a mutation in one of the receptors. You probably got the other allele normally functioning just fine, right? So you usually have to have mutations in both your tumor receptor genes before you start ignoring a signal, right? And just like oncogenes, you can have, too few growth inhibiting factors around, and that would, make the cell more likely to divide inappropriately. Or you could have receptors that don't recognize their signal. So even though there's inhibiting factors, if the receptor isn't recognizing those factors, then you might turn on inappropriate genes. You can also have transcription factors that aren't working and aren't turning on the right inhibition proteins. So this is all going to work in negative fashion, but it can work at each level just like oncogenes were. So also there are genes that control apoptosis, right? Like I said before, a lot of cells are kind of on the verge of going into apoptosis, and if you aren't sending in active signals to stop that mechanism, if those positive interactions with the other cells aren't around, the cell will just undergo apoptosis, right? So if you've mutated those cells, if you've mutated those genes that normally take a cell through apoptosis, then if you get in the wrong area, you're now immune to that self-killing nature, right? Yeah.
So tumor repressor genes, they can cause cancer by not doing their job, while oncogenes.
Yeah. Doing their job too well.
Okay.
Yeah. Some instances of these guys. The two famous genes, the breast cancer 1, breast cancer 2 genes, are both tumor suppressor genes. Certain cancers have a tendency to have different of these genes mutated, right? There's lots of these that have the potential for overstimulating cells or repressing cells, and certain cell types are more or less likely to use these other ones. So commonly in breast cancer cells, these tumor suppressor genes are mutated. Retinoblastoma, P53, these are tumor suppressor genes that are found in all kinds of cancer cells. So those are very, very common. Most cancers are going to have a mutation in P53 or retinoblastoma, including breast cancers, right? So most of the time, like I said, you have to have mutations in more than one gene to cause cancer. So these guys are very often have to be mutated before a cancer will be able to develop. Right? They're kind of like really fundamental control levels. So if you've got a good P53 gene around, then you might have other lesions that would make you susceptible to cancer. But if you got P53, then you're kind of sunk, right? If you got two bad copies of P53. So these have kind of more or less influence, right? Some are worse news than others. Tumor suppressor genes. Any questions about these guys?
So it's when the genes stop working, or is when?
Right. So a tumor suppressor, its normal function is to stop the cell from dividing. So if you've got one bad tumor suppressor, that's probably okay, because the other one's still keeping the cell from dividing. But if you've mutated both alleles of the tumor suppressor, now that gene's no longer functioning to suppress your cell division. Now you might have some other good tumor suppressor genes still going on, and you might not become a cancer yet, but it's kind of an addition strategy, right? You're gonna have to have more than one. Mutator genes. We've known about tumor suppressors and oncogenes for a long time. Mutator genes are probably in the last maybe 10 or 20 years that we've been studying mutator genes and the role of DNA repair in cancer. So genes that identify or repair DNA damage, these we call mutator genes, So a normal functioning mutator gene might be something like one of those damage-specific polymerases. Remember when we were talking about the difference between prokaryotic and eukaryotic polymerases? And there was like, dozen of eukaryotic polymerases. A lot of those are specifically just turned on to repair damaged DNA. And so the other polymerases are not capable of repairing the damage. So one of those guys would be a mutator gene, right? If you no longer make that functional polymerase, you might have DNA damage from some environmental stimulus, right? Like you might have had viral infection or inflammation because of a bacterial infection or something that stressed the cell, it caused the DNA to be damaged. Well, if you still have that functional DNA damage polymerase, it would get turned on, it goes through, screens the genome, fixes the mutation, fixes the DNA damage, and so your cell might be fine. But if you've mutated that, if that's no longer a functional damage control or damage repair mechanism, well, now I've got the chance that whatever that stress was, that viral infection, or maybe you got a sunburn and you got UV exposure and that damaged your DNA, you've got an increased chance now of getting a mutation in one of your oncogenes or one of your tumor suppressor genes if this guy is not doing his job, right? So this is like a higher level working. These guys are making sure that DNA doesn't get mutated. If these aren't working, then you have more chance to get mutations. And if those mutations are in oncogenes or tumor suppressor genes, then you might have a higher proclivity of having cancer. So these ones themselves are not going to trigger the cell to divide inappropriately usually, right? They're just the underlying mechanism to try and keep your DNA healthy. So this could be those DNA-specific polymerases. It could be that nucleotide direct reversal mechanisms. So if the DNA got methylated, a nucleotide got methylated and it looks like the wrong nucleotide, there's proteins that go in, clip out that wrong nucleotide and put in the right one. That's the excision repair. Right? If you've got the inappropriate nucleotide, those proteins go in, remove that nucleotide, and stick the right one in. Or if the nucleotide was just slightly damaged, nucleotide reversal proteins would come in. They would just repair the damage on the nucleotide. Right? You might not have to completely pull it out and put a new one in. You might just be able to fix it. So these are 3 levels of mutator genes, just mechanisms by which the cell can repair damage. So you might get a mutation in a proto-oncogene, but if the repair enzymes are working, well, you just fix the mutation and yay, you don't get a cancer, right? It's good to have a good mechanic, right? Keeps your car going. And I don't know, I throw up this illustration just to show you that we know lots of genes that are involved in this. Don't worry about don't worry about any of that. That's just a pretty picture, right? Yeah. So for your final, you need to make a poster of every possible. I love you, Matt Cruzen. All right. So we know the 3 types of genes that are involved in giving you an increased susceptibility to cancer, right? Having a mutation in any one of these may not be sufficient to give you a cancer. You might have to have lots of them. So in the case of a tumor suppressor gene, if you have a history of cancer in your family, it might be that you are inheriting some defective tumor suppressor genes, right? So here would be, you're a diploid organism. This would be two chromosomes, one from mom, one from dad. A normal healthy individual, normal wild type individual would have 2 good copies, and those are the black ones, right? So I got a fine normal individual, 2 wild type good copies of this tumor suppressor gene. Right? Well, somewhere in this cell's life, could be months, could be years, you might accumulate a mutation in a tumor suppressor gene. That's no big deal, right? Tumor suppressor genes act in a negative fashion. So if you've got work in a positive fashion to keep the cell from turning off. So if you have a mutation in it, that's fine, right? The mutant version isn't going to give you cancer because you got a good version around that's still suppressing tumor growth, right? So you might get a sunburn, you might damage DNA in your cells. If you've only damaged one of your tumor suppressor alleles, then you got a good one, you're fine. Well, if you go get another sunburn and damage more DNA, you might get 2 mutant copies of that tumor suppressor gene. That doesn't mean you're going to get a cancer, right? Because tumor suppressor genes are just suppress cells from abnormally dividing. But if they don't get any signal to abnormally divide, then there's no need for the tumor suppressor to be around, right? So it could be months to years, or maybe never, that cell, even though it's got 2 bad copies of the tumor suppressor gene, maybe it never gets any trigger to inappropriately divide, and so there's never any need for that tumor suppressor to keep it from dividing, right? So it could be months or years or never that you ever actually develop a cancer. What this is called is, well, maybe it should be called the 3-hit hypothesis, but it's called the 2-hit hypothesis. You have to have 2 independent mutation events before you ever have susceptibility, right? Then you'd have to have like a 3rd event that actually triggered the cell to divide, and now you're in a bad spot because now you don't have this ability to suppress growth anymore, okay?
Is that the suppression of growth specific to the region, so skin in this case?
Yeah.
In the sunburn?
Yeah.
Okay.
So it means, like I said, certain cells express different tumor suppressor genes, or they're more active in certain cell types. So if you inherit, or if cancer runs in your family, it's not usually like my grandma had cancer, so my mom had cancer, so I'm going to have cancer, right? It's usually a susceptibility for having it, right? In the case of tumor suppressors, your family history, you might have inherited a mutated allele of that tumor suppressor gene, right? So now you're more susceptible, right? If you have a single sunburn, a single, damage to your DNA, you only have to have 1 hit before you become susceptible to cancer, right? And then if you have some inappropriate trigger, you are just one step fewer from a normal, healthy person from getting cancer, right? But you could inherit a bad allele, you could have inherited 2 bad alleles, but you still might not ever get a cancer, right? It's just an increased susceptibility. It's a percentage game, right? How likely is it that your cell's going to respond appropriately? So you never typically inherit. Dominant cancer inheritance is usually very rare, right? Yep.
Sorry, I feel like you said this, but do you always need tumor suppressor genes? Or if the cell growth is regulating itself, is tumor suppressor only when it sees that it's going unregulated, unchecked?
Well, I mean, a tumor suppressor would be like the receptor in a cell that's hooking up to another cell, right? So 2 cells in your kidney sticking together. If that sticking together represses cell growth, then that receptor is a tumor suppressor, right? So cells always have tumor suppressor genes in them, right?
Okay.
So if that's mutated and no longer hooking up together, normally 2 cells binding together, right, making cell-cell contact, those receptors recognize I'm stuck to another cell, and that feedback to the cell saying we shouldn't divide. We're in our right location, we're hooked up to the right cells around us, there's no need to divide, right? If that's mutated though, it might still be there and still might be hooking up, it's just not telling the cell to not divide, right? So you might actually start dividing inappropriately even though you're hooked up to the correct neighbor cell. Or if you actually got pulled out of your tissue, it doesn't care whether it was still hooked up to a cell, right? That feedback wasn't there, right? So tumor suppressor genes, calling them that tends to make you think that, oh, they're only involved in repressing cancer, right? No, they're doing normal cell functions, like keeping cells stuck together and regulating division rates and stuff. And it's when they're mutated, we would say that they're mutated and they would be susceptible to a cancer when it's the feedback mechanism of their function that's been lost, right? But it might still be a fully functional cell adhesion molecule, and it might still be sticking very well, right? And what's mutated might be that downstream effect that it has of telling the cell not to divide, right? So when we say it's a mutation in a tumor suppressor, there are certain mutations, you could have a mutation in it that didn't affect its role in inhibiting growth, right? Proteins are multifunctional things, and they've got multiple domains, and they do all different functions. So it might be carrying a perfectly normal cell function and just be mutant for its activity in suppressing the the division, right? I don't know if I'm saying that well, but these are all normal, good genes that cells are using, right?
I get it.
Okay. I feel like I'm just repeating myself and not being any clearer. But if a cell does become cancerous and it starts dividing inappropriately and it starts migrating over the body, it's really important to know where that cell came from, because different tissues respond to different types of therapy, different type of treatment. There's lots of ways to treat cancer, right? You could just go in and surgically remove it, right? If that was a benign tumor and it wasn't growing very fast, you knew it was just growing in that tissue where you identified it, then you can go in and just surgically remove it. If you make sure that you got all the cancer cells, it's called getting clear margins. If you just remove all of the cancer cells and you know that you didn't leave any cancer cells behind, then that might be sufficient to treat the cancer, right? You just pluck out the cells and you're done, right? But you might have to do more than that, right? You might have to do surgery, remove it, and you may not be sure that you actually got all the cancer cells. At that point, you might need to undergo a chemotherapy, right? Chemotherapy is just going to be basically kill every cell that's currently dividing, which obviously is a very toxic thing to do to your body, right? You need cells to be dividing to replace cells that you're losing, right? So when you undergo chemotherapy, you're going to kill off all of your red blood, all of the cells that are dividing in your blood. That's going to be one of the first things that goes down, right? It's your ability to renew your red blood cells and your white blood cells. So you become anemic, you are really susceptible to infection because you're not making any white blood cells. You lose your hair because your hair follicles are continuing to divide, and if you stop them from dividing, your hair falls out. Lots of bad effects of that. We're getting better with chemotherapies. We're actually being able to make more specific molecules that inhibit only certain cell types to die. So we could treat, so the early chemotherapy is basically just every cell that was dividing in your body stopped dividing. And if that's a tumor cell and it's not hooked up to very good vascularization, hopefully you're just going to kill off all the cancer cells. So you stop every cell in the body from dividing for a short period of time, just to the point where you're really making the person susceptible to death. I mean, if you go on a chemotherapy for too long, you're gonna die, right? It's inhibiting any of your cells from dividing. So you put yourself on a regimen. We put you on a chemotherapy for a short amount of time, inhibit all cell growth. Hopefully that will stall the cancer, give the cells time to recognize, oh, I'm an inappropriate cell, and to apoptose or to senesce or to just die, right? And then you go off the chemotherapy and let the person recover. Right, get their blood cells, red blood cells, white blood cells going again, and then put them on another regime, right? So you bring this person to this point of crisis and then back them off, and then crisis and back them off to try and kill off all the cancer cells. We're getting better at this. We've got chemotherapies that specifically target certain cell types. So if you have a certain type of cancer, we can give you a certain type of chemotherapy, and it will only inhibit those cells to divide, right? So sometimes you can be on a chemotherapy, you keep your hair, you keep your red blood cells, and that chemotherapy targets only certain tissues, the tissue type, hopefully, that you've got a cancer in, and so we can be more specific to target certain ones. But that becomes really important to know what that cell type is, though, to correctly identify the right type of chemotherapy molecule to use. Right? If you've got a lung cancer, then we might need to use one type of chemotherapy agent. If you've got a pancreatic cancer, we might have to use a different type of chemotherapy. If you have a pancreatic cancer cell, though, that has migrated into the lung, you might be tempted to think that you have a lung cancer. But you wouldn't want to treat with lung cancer chemotherapy. You'd want to treat with pancreatic chemotherapy treatment, right? So identifying the cell type of origin is actually really important now in cancer treatments. I told you that story about my friend John Lamute, right, who had he had a tumor in his lung, but it wasn't. It was more like a leukemia, so they treated it according to leukemia, and so he's in remission. But knowing where the cell came from, not where it's currently growing, right? If it's metastasized, you could have a skin cell growing in the brain, or that's unlikely, but you can imagine that, and go to some very foreign tissue other than its origin, right? We can also do radiations, right? Kill off cells with radiation. And different regimens of treatment, right? You might do surgery and then chemotherapy and then follow up with radiation, or do a certain number of times of chemotherapy. Different cells respond differently. So this is the difficulty. Once a cell has metastasized, how do you identify it? How do you know it's in that, what its tissue of origin is? Yeah?
So do you like suggest that we should get like checked up once in a while?
Yeah, no, you should definitely get regular physicals every year, and if there's something inappropriately growing in your body, you should go get it checked out. Yeah. Yeah. A lot of it's mostly internal, and you would never know, right?
Right. So
Yeah.
do you suggest like people getting CAT scans?
A CAT scan isn't going to do much for you, because the tumor's going to have to be really big. No, but I'm glad you asked the question, though, because it actually leads into my next thing. Like, how do you know you've got a cancer, right? You might have inappropriately growing cells and not know it until they've gotten to be too big, right? One of the ways they scan for these is based on this phenomenon called the Warburg Effect. Otis Warburg, or Otto, I think it was Otto Warburg, was a cell biologist, developmental biologist in like the early 1900s up until like 1930. He did a lot of work on cell metabolism and cell respiration. And he identified, well, he was one of the guys that kind of led to this idea, but he studied tumor cells and studied their metabolism. And what we found is, and I'm not sure if he found this or if we just named it after him in honor, but most cancer cells predominantly use glycolysis for their energy production. They usually use glucose as their main energy source, and they don't usually metabolize fats, and they don't usually metabolize proteins. That's partially because they don't have very good access to circulatory systems, right? They don't have the access to the big LDL and HDL molecules that are roaming around your blood, depositing fats into the cells to grow. So they use whatever source is most plentiful, and so that's usually glucose. So this gives you kind of a little bit of a handle. If you can identify cells in your body that are using glucose more than other cells in your body, you might be able to identify cancerous cells before they get too big, before they become a tumor, right? So, Mark, you probably don't have to like go regularly and get one of these scans to see if you've got inappropriate glucose metabolism. But if you do have a cancer, like if they find skin cancer, what they will often do is, well, that skin cancer is fairly localized. They might just burn it off. They might cut it out and remove it. But how do we know that skin cancer hadn't actually metastasized and is now roaming around your body, right? So usually people will go and do follow-up exams. And what they look like is this. It's called a positron emission tomography. Basically, they label a glucose molecule, or a glucose molecule analog, so it kind of looks like glucose. But it's labeled so that we can see it under a detector. So you basically drink this sugary drink with this glucose analog, and we look to see what tissues in your body like suck it up and start metabolizing it really rapidly. So you can do a whole scan on a person. Okay, so here's a CT scan. This is a computed tomography, right? Kind of like a CAT scan. So it's showing density, right? Density of the bones here. This is the PET scan. This is the positron emission tomography. This is showing what cells in the body, what tissues in the body took up that glucose analog in high amounts. Now, what you'll see is brain, right? He doesn't have brain cancer. Your brain almost always exclusively uses glucose as its energy source, right? So if you've got a brain tumor, this isn't going to tell you it, right? Your whole brain is always going to look really densely stained here. And look, you can even see the canals. What are those called? The ventricles in the brain, yeah. So here's the dense brain matter, all using lots of glucose. Here, what do you think that tissue is? This one right here? That's the bladder, yeah. It's not bladder cancer. This glucose analog is filtered by the kidneys, and so it will concentrate in the bladder, but that's just normal. Where this is showing up, though, is liver. And it's also showing up here in the leg muscle tissue. Here's some more liver. This is actually an individual who had a skin cancer, went in and had the skin cancer removed, but went up and did a follow-up PET scan, and they found that skin cancer, or maybe it was some other cancer, we don't quite know yet. We'll have to do some tests to see what the tissue of origin was. But he's got a metastasized cancer, right? It's all over in multiple places in the body. He does not have testicular cancer. This is a male. The testes normally are very active in metabolizing sugar as well, in metabolizing glucose. So spermatocytes use fructose specifically. So there's lots of it concentrated in the male testes. So yeah.
Could you like mistake a cancer or mistake diabetes for cancer?
Mistake diabetes for cancer?
Like if someone said like, oh yeah, you have diabetes, and then like that it's actually a cancer or something like that, because
no. So diabetes is the failure to bring in the sugar.
Yeah.
And so what you would see is the circulatory system would just have lots of glucose, but no one tissue would be pulling up all that glucose, right? So this is looking for tissues that have actually brought it up, right? The other bad thing is all this green up in here, it's all in his spine as well. This is actually a really late stage cancer. This is bad stuff. We're totally over time though.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.